Last but not least is young Adam Butterworth, who's done some beautiful work. And Adam's been tasked with telling us what other omics UK Biobank should consider. And Adam told me before he's 37, but I think you look like me. Adam, 32, actually. Anyway.
That's chronological age, not biological age. So I'm not going to tell you about polygenic risk scores. I'm not going to tell you about all other omics. I'm going to tell you about proteomics. And I don't speak for a particular company. I don't speak for a particular community. So I'm a genetic epidemiologist rather than a proteomicist. But I'll hopefully show you some data, which is why I think proteomics would be a good thing to do in UK Biobank, much as the previous things we just heard about, I think would also be great to do. You've seen various flavors of this slide in the last couple of presentations, but I think by overlaying some of these molecular layers in something like UK Biobank, there's lots of possibilities for understanding fundamental biology, disease etiology, risk prediction, drug target prioritization, etc. And I'll hopefully show you some examples of that. Sure. So we have a mini biobank by UK Biobank standards based in Cambridge of 50,000 people, and that's been, if you like, a good test bed for, do these assays scale? Do they work well in subsets of these individuals? So we haven't measured all these things in all 50,000 people, but you can see here how we're trying to bridge the gap between DNA and disease. There's the heritable portion of disease, but how does it act? What are the intermediate pathways that we might try and target to prevent diseases? And as I say, ultimately, I'd like to see all of these things in UK Biobank. Some of them we have already, like the blood cell traits that Nicole mentioned earlier. We've just heard about the Nightingale platform that's about to be done. But I think each of these layers could offer distinct and complementary advantages. I'm going to talk particularly about work we just published in the last couple of weeks using a particular platform called Soma which is one of a number of different proteomic platforms we tried, but I'm not saying we should necessarily do this particular one, but I'll tell you a bit about what we find and why we think it was useful. So first of all, why look at proteins full stop? Well, they're the drivers of biology. The DNA might carry the recipe, but it's really the proteins that are actually acting in biology and often dysregulated in disease. And of course, many of the drugs that we have, the medicines that we use, target at the protein level. So it's a very relevant domain of biology, I think. As we heard, many different assays are now sort of maturing and becoming high throughput enough to use, but also in very wide angle. And as we'll see, examples like the Soma or the Olink platform now give the possibility not to measure tens of biomarkers, as we heard, are about to be released, but maybe hundreds or even thousands of biomarkers. And again, when we think about the limited, precious UK Biobank samples, what we want to do with those, perhaps things that give you a lot of data points and a lot of potential answers might be the way to go. And of course, proteins are very relevant, therefore, to a wide set of diseases and phenotypes. So as we've heard in UK Biobank, there's almost any kind of disease one can imagine, so there's potentially a very wide relevance here. People sometimes say, well, in studies where you have RNA and DNA, why do you need proteins as well? Well, we think there's an imperfect correlation. Actually, this gives you another layer of detail, and we find things that you wouldn't see in those levels. So here's the assay we used. It's called Soma Scanner. It's from a tech company out in Boulder, Colorado. We tested 5 or 6 different assays at the time in a small pilot study, as we heard about with the whole genome sequencing, try and find the best thing to do. Ultimately, probably this won out because it was the broadest. And as you can see here, the company now does, in fact, I think more than 5,000 different proteins that they can measure in human plasma in a relatively scalable way. And that's using this aptamer-based approach, which I won't go into the details of for time. On the right here, you can see it's a very diverse set of proteins. So it's not focusing on a particular biological domain, although it's not a completely unbiased approach. So they have been selected to be ones that might be of particular interest. Before you run these assays in large scale, you often want to know, well, do they work? Are they reliable? In some ways, we viewed our experiment, which was essentially doing GWAS of 3,500 proteins in about 3,000 of our blood donors, as a genetic validation. So what you see here across the bottom is the position of a genetic variant on the human genome, chromosomes 1 to 22, and then we utilize the fact that each of the proteins that we're measuring is coded for by a gene somewhere in the genome. So on the y-axis here, you have the where is that gene that codes for the protein. And you can see this diagonal red line that runs through. These are what we call the cis associations. So often you do your genome-wide screen, you look at 10 million imputed variants across the whole human genome, and the only region that lights up is right where the gene is that codes for that protein. And that already tells me that that assay for that particular protein is picking up the right thing. So in some ways, we can use the genomics to tell us that we're measuring the right thing. The blue dots that you see, the sort of pale blue dots, are what we call trans. That's a region somewhere else in the genome that seems to be influencing this protein. And often these make good sense. So we might have that the gene is a receptor for the protein, or the genetic signal is near the gene that's the receptor for the protein. But much of the time, we don't know what these are. And these are kind of starting points for novel biological understanding. Why does this particular region of the genome influence levels of this particular protein? What you can see as well is that there are these kind of stacks, and these are some of the kind of very highly pleiotropic, some of the kind of fundamental regulators of biology, or perhaps areas that affect big protein cascades, like complement factor, for example. So we think there's lots to learn about biology. In our paper, which you can read about, just came out in Nature, that we found about 2,000 genotype protein associations. So something like that's about 4 times more than had been published from studies previously. And I was going to try and create a cartoon of what it might look like if you did this in 100,000 or 500,000. But I think the blue would be probably a bit overwhelming to see. So what can you do with this data? Well, actually, we have a relatively healthy cohort of blood donors. So unlike UK Biobank, we don't have many incident disease events at the moment, but others have used this platform in other studies to show that you could start with 1,000 proteins and boil them down with statistical reduction techniques to a 9-protein score. And they showed that you could actually kind of predict future cardiovascular events by dividing people into deciles of this score. So you can see the people here in the 10th decile have a much lower, a much higher chance of having a future event. And one of the proteins that was particularly interesting to us, the second strongest one, was this protein called matrix metalloproteinase 12, MMP12, and they showed that people who had higher levels of this had higher risk of events. What we did in our study, because we were looking at genetic data, is we tried to answer the question of, it might be a predictive biomarker, but a predictive protein, is it a causal protein for heart disease? So we looked at a series of about 14 different genetic variants in the MMP12 gene region that all independently affect your level of MMP12 protein. And surprisingly, we found that the alleles here that increase plasma MMP12 actually have give people a decreased risk of heart disease. So we have an interesting situation where you might have been quite excited, particularly if you're one of the companies who has an MMP12 inhibitor that people have thought about for preventing or treating lung disease. You might have thought this would be good for also preventing heart attacks. Actually, our data suggests that this is perhaps a protective biomarker for heart disease, perhaps produced in response to initial damage. So there's definitely ways we can find causal insights into disease, sometimes in surprising directions with relevance for therapeutic targets. The other example I'd like to show you is about this idea of bridging the gap between DNA and disease. The GWAS era for diseases has been phenomenally successful at finding statistical associations, very small p-values, regions of the genome that relate to disease, but often they look messy like this. This is a region where there's a signal with inflammatory bowel disease, and there's 30, 40 genes in the region. How do we know which one it is? Probably many biologists could make a good case for any number of these genes. One of our protein signals happens to look almost exactly the same, statistically co-localizes, and actually it was a cis signal for this protein with the gene highlighted here called MST1. This suggests that actually maybe the causal actor in this region is MST1. Actually, when we looked at our other proteins, we also found other proteins that shared almost exactly the same signal. If I flick backwards and forwards here, you can see the disease signal and the protein signals looking very similar in the same region. And this was for another protein that's coded somewhere else in the genome called BLIMP1. And actually, when we looked at the inflammatory bowel disease, big GWAS papers, there's also a hit in the gene region for PRDM1, which encodes BLIMP1. And again, previous studies had made good cases for both of the two genes in the neighborhood here as being the biological actor. We actually think our data suggests that this is PRDM1 in this region and MST1 in the other region. So we can start to fill in the layers between DNA and disease and start to try and pinpoint, go from beyond statistical associations for disease to some meaningful biology. Now, I could stand here and tell you proteomics is going to be a wonderful panacea and we should go out and do it immediately and it's all going to be fine. Of course, there are challenges along the way. Some of them are specific to proteomics, but of course, many of them are germane to many omics assays or even kind of smaller scale biomarker assays. We obviously want to validate these assays before we go out and do them at scale, and we obviously used assays that have been used in somewhat smaller studies than our study, and again, maybe studies like ours can act as the pilots, if you like, for a large-scale biomarkers in UK Biobank. There are always challenges of interpretation, whichever domain you look at. In proteins, we perhaps have challenges about whether a protein is free in the blood or is in a complex, and how we interpret those measurements and results. There's great potential for non-biological variation, batch effects, etc., to influence things that need to be carefully controlled for. And we know the UK Biobank team has great expertise now in doing this. And of course, by the time we get to going beyond one protein, one measurement at a time, how do we go from the 35 biomarkers that are about to be released to 3,500 proteins? That brings different challenges in terms of statistics, computation, bioinformatics, etc. So it may not be easy, but I think there's enormous value of doing these kind of things at scale. So just to conclude, I think certainly many omics assays, and particularly some of the multiplex proteomics assays, are now becoming sufficiently high throughput and reliable for use in very large population studies. And they're also particularly wide-angled now. They're really growing in the diversity. A few years ago, we were talking about panels of 10 or 30. Now we're talking about panels of hundreds or thousands. And that just enables us to answer a really greater range of questions. And of course, we shouldn't forget these challenges. So really just thank Ben Sun, who was the PhD student who led all the work on the proteomics, and also the diverse range of funders, both pharma, tech companies, as well as national and charity funders who enabled all of that study to come about, both with measurements, creating the study, the blood samples, etc. So really a big team effort, and perhaps some hints about how these things might be funded going forwards. Thank you.